Hi, Miss Patrick and all my friends. Welcome to Jerry and Jack's animation project. In this video, we're gonna briefly introduce telomeres, one of the greatest discoveries in genetics and biology. When hearing the word telomere, it might seem to be remote to you. However, it's highly relevant to our biology study in DNA replication.
In this video, we use Minecraft as a tool to visualize the functions and determinants of telomere. What you guys currently see is the initiation of DNA strands.
Firstly, let's go through the process of DNA replication in the video. We all know that we have a double helix of complementary strands. I know it's not a double helix, but you can just assume it's after the functioning of helices. And it is now a linear straight strand. According to the knowledge in our textbook, we know that in DNA replication process, we have leading and lagging strands.
We are currently showing the replication of the leading strand DNA. Since the directional movement of DNA polymerase here is from 5' to 3', the replication process could go smoothly from the right to the left. However, in lagging strands, we need the setup of primers to help this replication process.
Because of the existence of lagging strands, we will inevitably have Okazaki fragments. I'm not introducing the procedure of DNA replication and some knowledge of it. The point is, the last RNA primer in lagging strands has been put randomly. What I mean by this is sometimes RNA primer cannot be put exactly at the end of DNA strands by primase, and this actually will cause the last Okazaki fragment to not replicate all base pairs at the end of DNA strands. The awful consequence of it is you will lose base pairs after replication. This is serious since it will result in inaccurate expression of proteins, and it is unacceptable to our body because proteins are so important.
Unlike bacteria, human beings don't have circular DNA, though it is not exactly a circle. I mean, you can see that it's pretty ugly, so it's obviously non-circular because I drew it, so please don't mind that. But the meaning is that it can help us to have a brief overview. So theoretically, bacteria can proceed its replication without any impairment or loss of DNA because its shape is a circle, and this allows them to replicate in one direction only, so they don't have to use Okazaki fragments and more than one RNA primer in each replication process. However, let us go back to human. How could actually we prevent DNA damage from happening if our DNA is straight? I mean linear instead of a circle. It is the telomere which is the clear answer. The telomere is a DNA protein complex which works as a cap that binds to the ends of chromosomes. It has repetitive structure of TTAGGG, or you can say double TA and triple G, as a non-coding region. Now, we are no longer afraid of primers randomly putting RNA primer, and we are going to lose base pairs on telomeres only instead of those on coding regions, which means we are not losing the normal functions of cells since all the DNA that is lost is basically trash.
We now know telomeres used as protections to DNA. However, constant shortening of telomere will also incur some problems. According to scientists' research, shortening of telomere will induce the cellular apoptosis, senescence, and oncogenicity. Let's first talk about apoptosis and senescence. I have to make clear that though both apoptosis and senescence will result in death of cells, cells pass away in different ways. Apoptosis is a predetermined death through the activation of certain signaling pathway by physiological and pathological conditions to allow lysosomes to release digestive enzymes and then break down the cells. Here's a clip for the apoptosis. It's quite exciting. Senescence is not predetermined. It's a stable, irreversible growth arrest of cells. It relies on the genetics involved in aging mechanism and happens in a non-fixed period of time. Death of cells is realized through the inhibition of their entering into G1 phase of cell divisions, so they can no longer divide and they're just getting older and older and just die. So why do we spend time on senescence and apoptosis? The reason for this is both of them are directly or indirectly influenced by the shortening of telomeres. In other words, length of telomeres is the evaluative markers for whether apoptosis and senescence take place.
individuals with shorter telomeres seems to have a greater risk for development of lung, bladder, renal cell, gastrointestinal, and head and neck cancers. It is also reported that cancer cells have relatively high telomerase to maintain the length to sustain the cancerous divisions of cancer cells. Telomerase, shown by the sheets in different colors, is an enzyme which is of vital importance to maintain the length of telomeres. However, we're going to go deep inside these telomerase. The only thing we have to remember is that it only actively functions in fetal tissues, which means the tissues of a fetus and adult germ cells, and also inside the tumors where it kind of malfunctions. So back to our point, the cause and effective relationship between the shorter telomeres and the risk of having cancers has not been steadily formed. One relatively credible hypothesis is that shorter telomeres can sometimes induce genomic instability by mediating interchromosomal fusion, thus potentially stimulating the initiation of early stages cancers.
Now we are in the final section, the negative influence of unhealthy lifestyle on the rate of telomere shortening. Before we start, I must emphasize the common sense. Rate of telomere shortening may indicate the pace of aging. Everyone will inevitably lose telomeres after each time of cell divisions. So even healthy people will lose telomere at an acceptable rate. As shown in the video, we can have a clear look at it. When you have the accelerated rate of telomere shortening, you may age quickly, which results in decreasing lifespan. Here are some factors that have influence on rate of telomere shortening.
The first one is smoking. A study conducted in white blood cells of women indicates that telomeric DNA is lost at an average rate of 25.7 to 27.7 base pairs per year. Those who smoke 1 pack of cigarettes per day, when compared to normal people, usually lose 5 more pairs per year. Therefore, the telomere attrition caused by smoking 1 pack of cigarettes a day for a period of 4 years is then shockingly equivalent to 7.4 years of life. Consider how many years we live. In total, maybe it's just like less than 80 years, but now we have to lose 7.4 years of life. And that is merely the influence of telomere, so it's definitely a bad habit.
The second one is obesity. As you can see in the video, an obese panda is keep eating bamboo. I'm definitely sure if a panda were a human being, its telomere would have been very short. Why? Let me give you the explanation. Obesity is associated with the oxidative stress. A famous Japanese biologist, Furukawa, shows that BMI and the waist circumference positively correlate with the level of reactive oxygen species in blood and urine. This oxidative stress in human body can induce DNA damage and therefore expedite the shortening of telomeres. Telomeres in obese women have been shown to be significantly shorter than those in lean women of the same age group, according to a study conducted by Furukawa. The excessive loss of telomeres in obese individuals was calculated to be equivalent to 8.8 years of lifespan decrease in a period of 40 years, which is an effect which seems to be worse than smoking. And that's the reason why as a panda must have a very short telomere.
The third one is unhealthy diet. This is a controversial part because some studies presented may be to some extent contradicted to what people considered as a healthy diet. According to a study done by a famous biologist named Cassidy, telomere length negatively correlates with the intake of polyunsaturated fatty acids and protein, but positively correlates with the intake of fiber and carbohydrates. Yes, protein is also a factor that will cause a decrease in the length of telomeres. To be more specific, in the experiment conducted by Cassidy, reduction in protein content of foods by 40% leads to 15% of increase in lifespan of rats. Okay, so here I'm not encouraging you guys to be a vegan, because I'm not vegan, but science always surprises us. So I'm not sure who is correct. But however, please just choose the meal plan which best fits your body.